Hi, my name is Arya. I'm a senior in Columbia College studying biochemistry and computer science, and today I'd like to tell you a little bit about the work that I did this summer as a Laidlaw scholar at the Broad Institute in the Sabeti lab to uncover differences between Ebola virus variants. So let's get started. Ebola is one of the world's most deadly pathogens. It causes Ebola virus disease, which has a 65% case fatality rate and a variety of clinical symptoms. The drivers of pathogenesis and viral mutation are still unclear. In order to effectively combat the 30+ outbreaks that have occurred since 1976 and to effectively treat Ebola virus disease, it's of interest to try to understand these. A key question in Ebola virus research is how the virus adapts during infection. Though mutations occur randomly, which mutations persist and at what frequency during infection can tell us a lot about viral dynamics. Do the variants that persist aid in viral replication, infectivity, disease causation? What are the selective pressures involved in an infection? We undertook a natural history study of Ebola virus disease, infecting monkeys with Ebola and collecting tissue samples across the course of infection. Variants identified in this dataset cluster in the VP24 gene of Ebola, which is known for its role in mediating the host immune response to infection. More specifically, these variants were clustered in the 5' UTR or untranslated region of the gene. The role of UTRs is unclear in biology, however, they are suspected to be regulatory. I suspected that these 5' VP24 UTR variants may be adapted for translation, and so designed a functional testing strategy to evaluate this hypothesis, following the rules of the central dogma of biology. We found that these high-frequency 5' UTR variants were significantly upregulated for translational efficiency relative to the wild type. We see as great as 3-fold changes relative to the wild type, which is incredibly fascinating. An interesting incidental finding was that in a control construct, which had a deleted 5' UTR, the translation was upregulated 10-fold, suggesting an inhibitory role for the 5' UTR. In summary, we found that high-frequency 5' UTR variants in VP24 upregulate translation, and we developed a proof-of-concept functional analysis pipeline that can be used not just for Ebola, but for all sorts of viral variants. I'd like to thank all the people listed on this slide, everybody in the Sabeti lab, and the Laidlaw Foundation for supporting my research. Thanks so much, and hope to see you at the symposium.